Nicolás.
Muchas gracias, me agradece mucho de estar aquí. Hablo un poco de español, pero por la ciencia mi español no está bastante bueno, entonces voy a hablar en inglés, disculpe. Muchas gracias a la SUMISH por hacer esta invitación aquí, y me encanta estar aquí en Chile con mis amigos, y hay muchos científicos que me encanta de encontrar también. Y gracias a la embajada también de Francia por ayudarnos para viajar y todo. Entonces voy a cambiar por el inglés, que no está mi idioma natural también, pero para ciencia es un poco mejor. Hoy voy a hablar de... disculpe, sorry. So today I'm going to talk about bacterial engineering, although, is that okay? Do you hear me? Okay, right, technical things, okay. So today I'm going to talk about bacterial engineering, although I'm not microbiologist, okay? Maybe you understood through what say Nicolas, I'm working about in the fuzzy area of science, moving from mathematics, physics, real biology. I did experiment, not anymore, unfortunately, myself, but I'm very much in the ground of experimental biology by using many different tools, including mathematics, as you will maybe see a little bit, and computing as well. But the question today is, in our laboratory, what we want to do, I try to get the thing working, what we want to do is basically to set up new medical diagnosis. Actually, we are working in the field of health science, salud. What we want to do is not doing new drugs, it's to identify very early the disease of the patient in order to act very early, or act differently if we can identify the capacity of a patient to respond to a drug. Then we want to know it before giving the drug. And we want to know if a patient could be sick before being sick. So the challenge is big, because it's really the great challenge of the next few years which are coming here. It's about personalized medicine, how to change the practice and to act early or before the disease is coming. For doing that, we need to address very classical things. We need to understand molecular mechanism, okay? Otherwise we cannot do anything. But out of this molecular mechanism, we want to extract something which is called biomarker. There are parameters, quantity of proteins, or mutation in the genes, or a flux of a metabolite. There are quantities which are changing when there is something occurring in the body. So that's what we are doing, but when you identify that, then you want to do a diagnostic kit to look at it. So it could be an antibody, an immunoassay, but when the disease is complex, when the biomarkers are complex, you need more than an antibody. And that's why we are going through synthetic biology to do something a bit more complicated to fix complicated disease. So, very briefly, it's not the point of this talk, but you must know why we are doing synthetic biology in the context I'm going to show you. It's because every day we are going along this kind of chain of discovery. We talk with clinicians at the hospital. We collect samples from human, disease or not. And we look in this sample if the people got different parameters. So we do so-called omic approaches, proteomics, genomics, metabolomics, stuff like that. We do obviously a lot of mathematics to treat the signal, after treating the signal to do differential analysis. But after that, we don't stop there. What we do is we do dynamic molecular simulation and modeling, so-called system biology, to try to understand why this is moving. And if we understand, let's say we are successful, for instance, let's be happy, and then we got biomarkers. We identify and we understand why this molecule move. That's a, let's say, given situation. It's not always the case. But when you get here, then you do specific probes, like sandwich of antibodies. You do that with a robot. You put that in a clinical situation, and you're happy with that. Very often the diseases are more complex. When you treat about Alzheimer's disease, when you treat about cardiovascular disease, how to prevent an infarctus, heart attack, before it occurs, not after, before. How to prevent that. It's very complicated. So we have to combine all these data, very complex as you may know, with mathematics approaches we develop in the lab, and to try to combine that as well, not in a static way. Now the biology comes into the dynamic era, where everything which is important and tiny are dynamic. And so we should look at the dynamics of the mechanism, and that's what we do with the dynamic simulation. So the picture is a bit more complex day by day, and an additional complexity is the well-known chain of discovery for doing a new drug for the company or for a laboratory, but unfortunately it's so expensive that there are only companies doing new drugs, because any laboratory, academical laboratory in the world is not rich enough to put millions or billions of dollars or euro onto the table in order to reach the whole chain. So the academical research is over here, and then the real chain of drug discovery, unfortunately, is in the company area. But in the diagnostic, it wasn't the case. It was like artisanal, you know, because the market was small, the question of diagnosis was secondary. We want to save the patient first, and then we will see before predicting that. This is changing completely, because it was demonstrated that most of the drugs are not efficient enough, and they are not efficient enough, to make the story short, because they are not given at the right person. There are a lot of good drugs, but they are not given. They give the same drug to everybody. But if you give the right drug to each kind of person, then it's going to change the treatment without inventing new drug. And in addition, it's less costly to develop new drugs. So people became happy, and now the convergence of diagnostic discovery and drug discovery is called theranostics, or personalized medicine, meaning how to measure each patient individually, each individual parameters, to decide this will be the good treatment for you, this will be the good treatment for that. So we have to look at the parameters of each patient. And so now the new drug strategy needs diagnosis and combined diagnosis. It's so-called companion test, the diagnostic test companion to a drug discovery. Well, now why do I say that? It's because if you want to do a test, today doing a diagnostic test is very costly. If you want to do that every day, it's very, very expensive and very painful. Could you imagine yourself going to a laboratory to have a blood sample every day or every month, or each time you have a therapeutic decision? It's too much. It's expensive and it's painful for the patient. So we need to imagine a new way to look at the parameter of the patient. And here comes synthetic biology. So synthetic biology is a sort of almost new science, which is now something like 10 years old. In a way we know it now, because synthetic biology word was used in the 80s already. But it is a sort of affiliated with systems biology. Actually, we are doing the same thing when we do system biology and synthetic biology, but the other way around. When we do systems biology, and that's what we do for learning about mechanism, what we do is we try to use mathematics and experiments to combine the data and understand the system. When we do synthetic biology, we do mathematics and experiment to try to design a system. We don't need to understand it, we design it. But we want it working, which is another thing. So the tools are the same, and the expertise are the same. But what did we learn from systems biology? We learned that biology is much more complicated than in a book. Okay? It's multi-layers. But not only that, there are dynamics. And not only that, the dynamics at different levels are not the same. Well, we have a lot of work for, there is a lot of room for us, scientists. But as well, what we learn, and this is on the bacteria, for instance, so probably something you know better than me, but we had a big European project which gathered something like 120 laboratories in Europe, just for looking at Bacillus subtilis. Just that. And why? It's because we remeasure almost everything which was affordable right now in a dynamic way, solve in a time. So transcriptomic, metabolomic, proteomics, everything in the same day, the same place in Europe, and that was a mess. But it took us 4 years for doing that, with all the mathematics behind that, with all the statistics, with all the experiments, redundant experiments with different kind of technology, nibble genes, transcriptomics, and stuff like that, absolute quantitative proteomics. And what we had, just a few examples, what we had, it's not like in a book. The central carbon metabolism, for instance, something that we all know in the book at least. Here you recognize maybe the glycolysis and the TCA cycle. Here you see among the time, the expression of the enzyme, just among the time when you just shift from glucose to glucose plus malate. Okay, so if you just stop it, well, I cannot stop it right now, but if you just stop it, you see that the snapshot of the whole network is not the TCA cycle or the glycolysis. It's a part of it and a part of a TCA cycle. And not only that, I haven't drawn that here, but it was a bit complicated. But the relationship in between enzymes, there are some new ones we didn't know before. They are just snap dynamics interaction, snap small metabolized, secondary metabolism. Okay, so it was complicated, it is dynamic. Look here at the glycolysis and the same TCA cycle. Again, shift from glucose to malate. You have here in blue the proteomics of the enzyme. So here in green, the transcriptomic, the RNA, okay, the mRNA. And in red, the relative product on the enzyme. Everything measures the same day, the same time, among time, right? It was so-called big experiment. And what you can see, it's very often, it's like the biological book say, so okay, when you got increase of mRNA, increase of proteins, and sometimes increase of a product of metabolite, which is processed, but very often it's not the case. Look at here, it goes down, it goes up, but there are counterexamples where it goes, where is it? There are a couple of places where it goes down and the metabolite goes up. You may find it for me, or well, okay, I don't know where it is, find it. But you can see here, the protein is decreasing, the mRNA is increasing, and the metabolite is just having a peak. Okay, it's not that simple like in a book. It's all dynamics. And it's not easy, it's very difficult to predict, but when you measure this parameter, then you got the so-called parameter from the equation, from the model, then you can start to predict in a very fuzzy way what's going on. Okay, so the systems biology shows that the picture is complicated. Just look here at the correlation in between the abundance of the enzyme and the flux, the relative flux at the same place. When it's a big ball, that means you got a big flux. When you got a big abundancy, or the same abundance, well, let's say good relative abundancy in between the quantity of protein and enzyme and flux. So you can see that there are only here, that's a glucose to glucose plus malate. Here you got malate plus glucose. And you see it's not the same. Any change in the environment changed the picture of the network. Okay, I'm sure you knew that already, but it was difficult to demonstrate, or everybody knows that basically, but to know in which extent it is moving is complicated. And it's not only that, the complexity. The complexity as well shows that we just saw that when the context change, even one parameter of the context change, the whole picture is changing within the bacteria. But now we just discover in the last 10 years that our context is even more important than our own metabolism, which is about the microbiome. Okay? So very briefly, metagenomics shows that the role of microbiome in human disease, the gut microbiome influences diabetes, obesity, okay, we may understand the relationship, cardiovascular disease, colon cancers. But not only that, the people show that depending where you live, you don't have the same microbiome, and then you don't have the same risk of disease. Not depending on the correlation is much higher than your genetic. Okay, the correlation is higher with your gut microbiome compared to the disease, and the correlation is better with the microbiome compared to the genetics. So, but not only that, maybe it's not maybe. There are quite a few proof now in the literature that the microbiome can as well influence your own behavior. So here that's a paper about appetite, but there are on anxiety it was demonstrated, and many things. So there are a lot of things in literature. So the microbiome is a sort of controlling your environment of your own cell and changing the picture we all show with the Bacillus subtilis. So we are not going to see the Earth in the same way now. Although we don't know how to handle that, it's so complicated, but for sure we cannot say we didn't know. So we have to try to figure out how to work with that. And obviously the plan now for the next year, or the coming year right now, is not only to do systems biology, proteomics, transcriptome, and that, but additionally adding xenometabolome, microbiome, the environment change, the one you can measure, at least your own environment change, in order to put that in a picture and try to figure out, segregate the population of patients you are going to treat with one molecule or the other one, and maybe address personalized healthcare later on. So for doing that, you need to introduce new parameters. Just very quickly, because again, it's not the topic of the day, but there are multi-layers in biology, and there are multi-layers in pathology. There are pathology which are connected between them. You know, when you got a diabetes, you may have heart attack, renal kidney disease, and so on. And there are as well social relationships. Social relationships, geography, food, etc. change completely the fate of your own genetics. So everything like that, that was demonstrated. We don't know and we don't understand everything on that. We know that it is important. So now the picture is going to be even more complex. So at least Facebook is going to be useful for something. So now in our field, we want to address early what's going on in a patient, the earliest we can, as I was saying at the beginning. So what can we do? We have a clinical question. The clinician comes and asks us a question and say, we need a biomarker, we need a device to measure that, and then we screen, we diagnose, or we monitor patients. But the thing is, many things are changing because if we want to diagnose each patient, we need to do that in the easiest way. And comes into the field, the so-called point of care, POC, point of care. It's about not going to the laboratory to have a test, but doing that even yourself or in your own doctor consultation. But for that, doing just that, it's already complicated. You have to be reliable, sensitive, specific, doing that quick and not waiting for three hours with a finger like that, waiting for the reply and so on. So there are critical practical questions. And so the whole field is concentrating the connection in between biology and electronics, so-called biocapture, biosensors, and so on. We are into the field there, but we think that the future will be, for us, where the electronic itself will be replaced by biology. Let's say in the other way around, we want to play and we think we can do the work of the electronics here just by using molecule as an electronical device, or it's not electronic anymore, it's only biology. And the advantage of that is then everything is full biology, and then it's very much compatible with the body. Then it's not easy to put an electronic capture within the body, but maybe something biologic. I'm not saying alive, I say biologic. Maybe it will be easier. Then synthetic biology, so you probably heard about that, is really the based on synthetic biology. The fundamentals are, does biology is able to calculate like a computer or not? The answer is obviously yes, otherwise you are not able to calculate yourself. But having said that, can we use cell to calculate and do, okay, there is a very, very fundamental paper, 44 or 45 pages, a work made by the physician, theoretical physician, okay? And he did play with plates like that and bacteria, and he demonstrate like that bacteria growth motifs here were predictable by computer systems, and the bacteria are more efficient to, let's say, calculate NP complex. So that means a sort of algorithm that the computer needs a long time to calculate, and they do that spontaneously, much quicker and much precisely. And so he demonstrates that. It's biological paper. It's very interesting to read that. especially for students. The way he did that, it's amazing. But now if we want to engineer a bacteria, because you understood now we are going to try to play with this bacteria or with biological system in order to reprogram them for us in our lab, meaning we want to do system for diagnose, to measure parameters, but there are other people doing other things. But for doing that, it's like building a plane, the same things. Just to remind you that synthetic biology starts in the States in the Jet Propulsion Laboratory in MIT, the one which do the engines for planes and rockets, because it's about engineering. And in biology, in general, we are not very strong in engineering. We engineer things, but the theory about what is engineering, how to handle the engineering, complex one, when you play your life, like going to the plane, you don't want the plane falling down, then it's not that easy. We engineer molecule, up to now, we engineer a bit some bacteria for bacterial protein expression and so on, but now we come into an era where the engineering is going to be complex, and you're not doing that with your brain and your pipettes, you are going to do that with your pipettes and a computer. Then we need people which know biology, mathematics, and model. And for that, doing things, standardization, abstraction, decoupling, modularity, okay, that's not a word we use very much in biology, but they are very easy to understand. If we want to build a system, whatever it is, a radio or whatever, you need pieces, you need devices, a group of pieces which are doing the same thing, okay, like a microprocessor. There are millions of same things, but each time people is building a system, they are not redoing the microprocessor, they just use the device. And the system itself, the radio itself, okay? So in biology, then we identify parts, device, systems, we standardize them in order to exchange with the friends, the concurrents, and so on, because we cannot do it all ourselves, and so on. And then we work about decoupling and modularity, meaning we have a problem. In electronics, when you need a transistor, you put a transistor here on the circuit, you put the same transistor here in the circuit, they are working independently. If you need an enzyme in a system in biology, you put it in cytoplasm, you put the other one in cytoplasm, they are going to interact because they are moving, it's auto-organized, that's biology, that's life, okay? The property of life are not the property of to make it short, of electronics. So in electronics, it's an advantage, in biology, it's a problem, okay? If you need two transistors in biology, you need to design two different transistors, independent and so-called orthogonal, which are not interacting between themselves, which is almost never existing in biology. And so why people are doing synthetic biology? As I was saying, for many different purposes. There are people designing new materials, like nanotubes, but not only nanotubes, a lot of geometrical forms with properties, circuits on that, nanocircuits. Fuel, obviously, is probably well known. Different kind of fuel, but not the one we think. It's not to replace petrol. I discussed with a large company like Shell, Total, Petrobras. They don't care about replacing petrol because they say now we find new places with petrol, so it's not a problem. But they care about the chemistry of the petrol to transform it, and 0.1% of efficiency for an enzyme in the cracking, for instance, and the preparation of the petrol, then it's millions for them. So they want to optimize the systems, and so that's why they do synthetic biology. For the rest, we can play with that to do electricity too, accumulating ions and doing electricity. There are people working on that. I will show you what we can do on medical diagnosis. Obviously, doing new kind of drugs, we got sometimes before seeing that in our body, because as you may understand, before injecting something, reorganized not only genetically from scratch, we have sometimes to wait. In the food assessment, especially not for doing OGM food, GMO food, sorry, that's OGM is in French. GMO food, it's for doing food quality assessment. And it is a real problem right now in all our community. We eat something which are being industrialized. We don't really know the whole chain of industrialization. There are chemicals in it. There are pathogens in it. There are many things we don't want to see in it. And so you may address that in your own place at home, maybe before buying or in the supermarket. So something very cheap, maybe affordable, and so on. And the green chemistry, and obviously close to green chemistry, bioremediation is a great field for synthetic biology. Okay. It's interdisciplinary, you understood it, that physics, biology, computing, mathematics, and chemistry, obviously. So if you like all that, you're going to be happy. Well, I'll shift this one. There are a couple of, in biology at least, I'm going to concentrate in biology. In biology, there are a couple of groups working, there are only few there. There are hundreds of groups now in all over the world working different things, but trying to change a bacteria to attack cancer cell, trying to reuse plant and reprogram a plant to create bone, to grow on a bone scaffold, to create and to create calcium around this bone scaffold, and then create proteases, biological proteases and not chemical proteases, and so on and so on. But there are very interesting works going on. And as it was stated in this paper in 2011, synthetic biology tried to come in Earth. But as you may know, to have something working in Earth in a clinical environment on your own body, there are a lot of long chain for clinical assessment, verification, regulation, and so on. So it's not easy to put something in activity in a market or in a use in a hospital. It's always a long story to verify that everything is working properly. So there is no application right now existing from synthetic biology in clinics. But there are a lot of work in order to try to work on killing cancer drugs, regulating homeostasis. It works in a mouse, regulating homeostasis of uric acid by just having a retrofeedback using an artificial cell. But there is no application right now in medical diagnosis. Just to notice that, for instance, there are even people trying to change commensal bacteria strain in order to fight cholera bacteria infection, and to try to, using quorum sensing, you may be more expert than me, but using quorum sensing to try to really fight the infection immediately after having ingest the bacteria and having the early infection. So how are we going to proceed in order to do a biological biosensor program by us? The principle is very simple. You get 3 points. How to capture the signal you want to look at. Okay, that's a sensing system. Could be a receptor, like in biology. It could be a molecule going through the membrane and a promoter inside. Many ways to capture a signal. How to calculate with biology. I'll show you something later on. And how to provide an output. It could be light, could be electron, could be chemical produced in a medium, many things, fluorescence, whatever you want. So you can do that as well, with bacterial. Not only bacteria, there are people working with eukaryote cell as well for doing synthetic biology. But you can do it as well with a non-alive cell, and I will show you 2 examples, or at least one example, with a non-alive cell doing works like synthetic biology or like a bacteria. There are a lot of advantage, as you may understand, to work with synthetic biology for clinical diagnosis, but as well there are some drawbacks. Because it is compatible, for instance, with biology, then it could interact with the body as well, very easily. So you have to prevent that, and so you have to work with a compartment, something stable, something specific. And so it's not that easy. But the great thing, it is disposable, it's human compatible, and as well you can combine that with many, many other interfaces like electronics, physics, and so on. Well, I told you already that it was a sort of a segmentation between layers of building. We have protein genes, reaction pathways, cells, tissue, and so on. But we have to model that. Now, we are not designing things by just doing experiment. What we are doing is we design in silico in a computer, and then we test it in a computer, dynamically speaking, by doing simulation. And when we think it's close to work, or we expect to work, then we do the experiment. And then it's a loop going back to optimizing the system and so on. And so for that we need to do some model, but to model the biology is not easy. So from our experience from systems biology, the rough general model doesn't work and cannot predict anything, and we want to predict something working in real and not only doing a publication. So we spend a lot of time to learn and to try to understand what is a real function, because we want to design something which is doing a process, a function. The molecule, we don't care. We want to provide a process, a function. And so we work about what is a function, and if you go in any kind of database, function is a text. So that means nobody really clearly understand. You ask 3 experts of the same molecule to describe the function. They describe it properly, but with different words and not exactly the same function, because the functions in biology are complex. What we discovered a couple of years ago, in 2004, it's all the functions existing around the Earth on all the molecules are supported by a very small number of elementary bricks of action. And the combination of this brick of action can describe all the existing functions, and even more, obviously, because there are functions that are not existing. So I'm not going too much into detail, but now when we look at a network, protein network, we change our paradigm to see a protein network, to model it. It's not a network of molecules interacting between them. We don't care about the molecule. We want to know where are the process. And as you may know, the molecules have a different kind of domains. The domain can support different kind of functions. They can have different kind of small functions. So let's take this kind of network of 3 molecules interacting with them with different kind of pathways for function, metabolite or not. Then if we now remove the notion of molecule, we cut it, and we just keep the domain with a specific function. And we just redraw the network. Then the network here, in terms of function, is representing the same network, which is here in terms of protein interaction. But you can see the topology, so-called topology of the network, is not the same. You have to know that all the mathematicians working and calculating on the network, they work on the topology on the network. And we, as biologists, provide them with that. So it's a network of people checking hands, basically, interaction, physical interaction. It's not a network of people talking with them, having a hierarchical relationships, being the father of somebody. You see what I mean? It's this kind of more interesting position questions about role and function are not represented by only the interaction. And so we try to represent them differently. And again, to make the story short, for that it's a problem. You have to do all the 3D structure of the proteins, the enzyme, assign all the subdomain of the protein. So now there are a lot of ways to do that. And when we identify a subdomain, structural domain of the protein, assign on each domain, not on the molecule, where are the specific local function on each domain. So that means a lot of structure function annotation, which should have been done in a database, and probably in the future is going to be done on a large scale. But if you do the same on the same network, the work, structure here, and 3D fold, and the so-called basic element of action, our elementary brick of action, which actually are in sort of sequence for each function, sequence of brick of action, then you will redraw the same network there. So this network looks more complicated, but means exactly the same in terms of function. But when we calculate on that, where are this function which is doing this and that, we only work on function and not on molecule which can do a function. You know, it's a sort of direct calculation on function, and here it's a kind of indirect calculation of function. So that gave us a great advantage for designing system. So we introduced that in mathematics and computing. We create some specific simulator on 3D. With each point here is a real molecule. And actually, when you work with small things like bacteria or vesicule, I'm going to show you vesicule, you can count the molecule in it. You can afford it. It's not like in a tube. It's something like it's about hundreds of thousands and millions of molecule, representation of molecule for each concentration and so on. We talk about this kind of numbers. Now computer can afford that. So that means we are not calculating in population. We are with that. Sorry, it's in French. We calculated with so-called multi-agent. Each point, each molecule has a specific algorithm working alone within moving in a 3D area. So it's a software which we developed, which is called HSIM. So that's for simulation. Now we want to address this composability issue, and we need part. And for part, it's like for electronics, we need sensors, we need logic gate at the AND or XOR and all these things. Switch, like for switch on the light, oscillators, memories, and so on. So, but for that, we want to reuse them. We don't want to create each compound each time we need one compound. So we want to provide compound, robust, which can support a different kind of environments, because otherwise we have to redo it again. And we want to design them in a way that we can as well simulate quickly. So it's a sort of like reinventing electronics from the very beginning with molecule. And so we will have a game and role with molecule, where some molecules are going to be used for fuels, for killing the system when we want, for switch, timekeeper, and so on. That's the kind of role. And so we put all the molecules identified for this role. I'm going to give you a few examples in a database, so-called CompuBiotic-DB, of reusable, non-toxic molecule that we can put within a system and reorganize in a Genius network. But directly, it's already in the computer, so you can play with it. And so, for instance, an oscillator, maybe you know that in cyanobacteria, there are oscillators for 24 hours. You put this molecule in a tube. You put 3 molecules in a tube, and a few metabolites, and it oscillates with a period of 24 hours in your tube, without the system. So that's an enzymatic oscillator. We look at the detail of the structure and dynamics. It's very complicated. It's made. It's based on allosteric transition, very complex. So we model that in very much detail. And out of what we model, we managed to design a small, more simple system of oscillator where you see the name of the molecule is not made there, because you can change now. It's a system with molecule. Each molecule have different properties, which are already fixed. Okay. Each molecule have different properties we already fixed. They must work in this kind of network, but we can change the molecule. The only thing they have, it's they should accommodate the constraint we put on this molecule. So that's a way for doing now different kind of oscillator with different kind of enzyme, which oscillate. Maybe it's not easy to see, but it's red and yellow. It's oscillated. There are 2 oscillators in this system. As well, we want to capture signal which are sometimes outside of the cell, so the molecule can go through the membrane, but not always, obviously. So we work on re-engineering receptor by just grafting. So we formerly been a lab specialist of monoclonal antibody and engineering of antibodies and so on. So we want to engineer antibodies and here with a functionality of a receptor here. So that's another way to capture, and that's another way of capturing things. It's a fully chemical. It's a sort of cyclic peptide here on which there are 5 orthogonal sites, chemical orthogonal sites. It's very complicated, so there are 5 sites for different chemistry. So you can put 5 different things on the top of it using just chemistry. And so if you put the CDR from the antibodies, then you create a very, very tiny antibody which can go through the membrane. So that's a kind of chemistry you have to do. If you want to do a sort of remote control for your cell, it's possible. So for doing a remote control from your cell, you use cage molecule, like this kind of compound. Oops, sorry. My remote control is not working. Okay, so you kind of use this molecule which, when they bind, they change the conformations. And sometimes you can change the conformation of some specific bond made by the chemist just by using a laser. So you have a sort of laser which put a bit of energy in the system, shifting a bond. Then the piece of peptide, for instance, is going to be digested by the enzyme because the bond has shifted thanks to that. And it's going to be the shift and the switch for your system to start to work whenever you want. So you can even switch on cell from away. So it works quite well, this thing. And obviously, to calculate, we need logic gate and or XOR Boolean logic. So a couple of years ago, Wilner showed that it was a chemist. He showed that with a few enzymes, with 2 enzymes, it was possible for of doing AND and OR and XOR gates. Maybe if you are not if you are not confident with the gates, means you have an input. For instance, here that's an AND. When you got nothing and nothing, you got nothing and nothing means nothing. Nothing and something, you get something. Something and nothing, you got something. Something and something, you got something. Okay? So you got 0, 0, 0, and 1. When it's a OR or XOR, that's a different pattern. Okay? It's just a matter of pattern, just of logic reply. So he designed that. It took him 3 years of designing these 3 gates. Okay, so we cannot afford that. If we need each different gate because of the composability problem, if you need 10 gates, you need 10 different gates for doing a circuit and a small circuit. So we work to try to understand what kind of combination of enzyme and metabolite can, by combination, provide an AND gate, OR gate, NOT A gate, and all the other one. And so we came up with this kind of pattern. There are many others. I'm not going too much into detail, but if you want in a discussion, we can discuss about that. But with all this pattern, we transform that in a computing environment, in a system, in rules, mathematical rules, which now we use to look at the existing network in nature. And so because we have patterns of AND and OR, we look at all the AND and OR in networks in biology. And then we have now thousands and thousands of AND networks, but we have to verify if they work. So that's what we do experimentally. Here, again, that's molecule that's from E. coli, a sort of extract from central carbon metabolism, but not only. There are pentose phosphates and a bit of a central metabolism of E. coli. And you see here some lines. So there are pieces of networks which, when they are extracted and just put into a tube, they are doing here AND, here OR, etc., etc. So within the same network of something like 45 enzymes, we can find something like 78 different gates that you can extract and which are robust in a tube. So we can extract that and put that in our database and play with that with our software to design a system and then see and look if we can do a circuit. So they are validated experimentally. You see a couple of examples here of different gates which are ANDs, OR, but with different enzyme and metabolite. And they are all been validated experimentally speaking. Okay. So now with our rules and software, we can extract out of the biology of all the organisms which come every day in the database, new set of logic gates. Can we combine them as a circuit? And you see now the early circuit we did with 3 gates and 4 gates, they work perfectly because they are very robust. Even out of the environment, this obviously you have to be careful about the link. We don't have the cable, electric cable. It doesn't work in biology, unfortunately. So you have to think about how to plug different circuits. That's the main game. But otherwise, it works perfectly. So we can concatenate or doing some circuit with enzyme. So now we want to design it, but not by hand. We have that in a database. We don't want to learn all the database by heart and say, oh, maybe I can put that here. That's what we used to do in the past, and it's too long. So we designed a software which helps us to address this part from the design to the simulation, for the design and the simulation of the system. And after, obviously, there is a experimental validation. So the software is called BioNetCAD. It's a tool. It's a NetCAD, computer-assisted design tool for synthetic biological design. How does it work? Very briefly, there is an interface, graphical interface. When you choose, I take an enzyme, I take a metabolite, and I want to draw my networks with name, generic name, generic protein. This is a receptor, this is an ion channel. When you say that, it's already meaning something. Ion channel, receptor is not just only an enzyme. There are some constraints behind that. The software is already identifying what you are doing. And so you accumulate the things, you can play with it. The students are doing that very easily. But behind that, what the system is doing, its cell designer is an interface, graphical interface, is looking at what you are designing with the system, BioNetCAD, and picking up the molecule we put in a database, okay, ready to use for synthetic biology. And then it's going to, with your circuit, run simulation, different kind of simulation. I'm not going too much into detail. And then you look at the behavior. If you're not happy, you can go back and redesign. But you look at the system working in your computer. When you are happy with the behavior of your system, then you may go to the experiment. So how does it work? It works with an early abstract network, a network with no name for the protein. Just this is a receptor, this is an enzyme, this is a metabolite. And then this abstract network with a sort of a loop like that of implementation is going step by step to be filled by the protein from the database, which fit with a constraint. Okay? A molecule, an enzyme which cut another molecule in 2 pieces, it's something we can find in the database. But it's not only a protease, it could be a DNase. But the substrate is not the same. So you see, by just saying that, you see that already the software is just catching up the constraint you put just by saying simple things. If you say DNase, it's already, the substrate is a DNA. That simple thing like that, accumulating at the scale of the networks, is very useful. And at the end, you can run simulation. And obviously, a same abstract network can end up with different real network with real molecule doing the same work. Here, it's a logic gate. And then you can see how does it look like for simulation. Classical ordinary differential equation here. Non-classical one, multi-agent-like system. So that means individual and 3D simulation. And here, that's our experiment. Obviously, it doesn't fit exactly. But all the concentration here are the right order. That the only discrepancy are for the numbers. But that's the same scale of time, and etc., etc. So it's not that bad. It's not that bad, just to be short, because the system is small. Okay, just to make it real. When the system is big, the simulation are not that close to the experiment. So it's not because we work better than the others. The system is simple. Okay, is it how does it feel when you perturbate it? So we play with pH. Obviously, you see that's one of the gate with 1 point. Look at the perturbation for pH. Obviously, the enzymes are sensitive to pH. So that means you have to take care of the pH of the system, depending the enzyme you are using. If you change the enzyme, you are not having the same dependency. But look at the albumin. No albumin in a system. You put albumin in the system. Whatever the concentration is, the perturbation is the same. So it's a kind of entropic perturbation more than specific molecular perturbation. And then you reach a point where, with a given concentration, it's even more perturbating, but again, it's a plateau. So it's more an entropic perturbation made probably something with the non-specific interaction of albumin, which is well known for that, is interacting with almost every molecule, but non-specifically. So that means you reduce the dynamic of the system. So now, how do we transform that? It's something we can use in a real environment, at least in a real condition for disease. So, as I was saying, we want to shift from the classical immunoassay, ELISA's test, for instance, or Bandlet. We want to shift out of that, and we want to do something very specific, look at few and different biomarkers at the same time. Now, there is no disease where you look at one biomarker, and it's enough. The diseases are so complex that you need 1, 2, 3, sometimes 10 different biomarkers. So your system should be able not only to show you one color here, it should be able to test 1 or 5 biomarkers, calculate this one is increasing, this one is decreasing, this one is absent, and so on. You have an algorithm to say, when this happened, this happened, this happened, then there is a disease possible. And then you put the message in a system. That's what we want to do. And obviously, if you want to do it on the patient, you can pick up some blood, but it's painful for the patient, for me at least. And you need to do that by professional, usually. Even for the finger, I tell you, in the diabetes, people, after ages, after years of testing them on the finger, they're really tired about doing that. And sometimes they stop to doing that. It's not because it's harmful, but every day, every year, it's harmful. So we want, as you understood, to look at the toilets in urines, and in the urines, we look very much at the urine since a couple of years. There are a lot of parameters coming from the serum, not all the serum, but coming from the serum, and which are very rich with your own parameters. So just by the urine in the toilet, we got a lot of things and resources to be happy for us. So the system would be whatever it is, a bacteria or a vesicule, artificial vesicule, a system with receptor, something which integrates the signal of the receptor, and something which puts some coloration or fluorescence, whatever you want. Okay, I showed that already. So the system would be designed, the system would be, for instance, here, 2 receptors using the same substrate, okay? But the substrate need to be produced by it's a product of this enzyme, and you need this substrate. So this enzyme is not working unless you put in the middle something to say you can start. It could be light, it could be chemical, whatever, so that the switch on. You switch on, this produces the substrate, but doesn't work if there is no biomarkers. If one of the biomarkers here is blue, the other one is yellow. If there are two, it's a combination of the color. For instance, it's something very simple. But you need to stop it at a certain time. So you put a time clock, and so that's an enzyme here where you know perfectly the turnover. You put a given quantity of substrate here. Here the substrate will be a consumer of the substrate produced here. And then at the end of the day, you will consume all that, and the system is going to stop after a given time. Okay? So you switch on, you stop it here, and it works if there is that. So you see, a very simple thing, it's not alive. It's just biochemistry within a vesicle. So obviously for that, you need to design it, simulate it, and then going to reality, and that's where it is really difficult. So I'll give you an example of cell-free one, and then a bacterial one. Cell-free means it's a liposome. Instead of having all these genetics, which are complicating, regulation and everything, okay? If we want to do something simple, like a calculation tool, I don't know how to say that, calculate in French? Calculadora, you know, the one for saying plus, minus, that's it. You don't need the whole system with the computer. You can do it with your computer, but you don't need something sophisticated. So if we want to know if there is this one is increasing, this one not, is that enough? We can do something simple. We don't need the whole machinery of a cell. There are some advantages and disadvantages. So for doing that, I'm not going much into detail, but we can discuss that there are different ways of doing liposome. But we want all our liposome the same, and it's not easy. It's not easy at all with all the system well known. It's very easy to do liposome, but with liposome with the same concentration of everything in it, it's not easy at all. Okay? So for doing that, fortunately, there is microfluidics, and microfluidics may provide you a very good homogeneity of the compound within the liposome. If some of you want to do some liposome or droplets, whatever, there are a lot of technology. If you want to do something homogeneous, forget about doing something else. We spend time, and our colleagues too, and this is the only way. From now. But at least it works well. And what we do is we simulate it. Obviously, the volume of that is so tiny. It's few nanomole, nanometer square cube. Oh, I'm going to go. So then the number of molecules in red, maybe you can see the small red point, is the one we expect in terms of concentration. So the number of molecules are simulated by the system here. You can see the curve here. That's what we see when you look at the tube. Here we can look at individual molecular system. It's just a simulation. And then we build it with, for instance, here, real life. We want to look in the urines when you got high glucose and high nitric oxide species, then you can suspect a diabetes. High glucose, one day it could happen, okay? You drink, you go to McDonald's and you drink a lot, you eat sugars. You could have a glucose one time the day you go to the doctor, okay? But if you got that plus nitric oxide, then it's not the same metabolite, and there is something going on. And if you repeat that, actually that's a protocol, even with the blood sample, they have to be repeated during months to state that you are diabetic or not. So if you repeat that, then you have to go many times to the doctor. I don't like that, so we try to do that in another way. So this system is just glucose. We manipulate the membrane in order to leave the glucose going through the membrane in a passive way. You can put a receptor or a transporter, obviously, GLUT1, GLUT4, etc. But we change the composition of the membrane in order to leave the glucose going through the membrane in a passive way. Then it is processed, and it is combined with another process, which is this way, which is looking at all the different species of nitric oxide at different level with different enzymes. And the combination of the two are made by NAD plus and NADH plus, which is basically where the the two mechanisms are crossing and are connecting with them to make the end. And then this compound is fluorescent, and we look at the fluorescence of the beads. Okay? That's a very simple way with a non-alive system. You can drop that in the urine, it's very stable, and it is just changing the fluorescence of the urine or changing the color of the urine by if you have high level of glucose and high level of nitric oxide. So that's obviously if you change the ratio with the simulation, we can look at the where we want the threshold, okay, to fit with the clinician needs, okay. But obviously, from now we produce the vesicle into the microfluidic system, and here you got an example of fluorescent vesicle. That's the same picture with a fluorescent vesicle having this measure of glucose and nitric oxide. Now we're going to do the same work with a bacteria. The problem with the vesicle, it's a vesicle starting from scratch never works, and you make it working, and it's very difficult to start from scratch in biology and make something even simple working properly. The good thing with bacteria, they work. They like that. They work, they are alive, they multiply and everything. But you won't have them working as you want, and this is the tricky thing. So the problem is completely different. With the bacteria, it works, and you want to control it. With our system, we control the composition, and we want to have it working properly. So the question is different. So we have a collaboration with a French guy which actually joined our lab now, which recently in a Drew lab. So we have a partnership with Stanford, and Jérôme published very recently this year a system in a bacteria which allow to do logic and memory, sort of memory stick, you know, USB memory stick. And the system is you use two recombinase under the control of two different promoters. Okay. The promoters which are looking at your compound. So that will be the sensors. So sensor 1, sensor 2. The biomarker is supposed to activate the promoter. Then these two recombinase are going to control the terminator here. And here you got the output, which is a GFP, for instance. Okay? And if you have this one, then the terminator is going to be turned, and this is going to read. If you have this one only, it's going to be turned, this is going to read. If you have the two of them, both of them are going to turn, and they are going to read. So that's an AND gate, which is done there. Now, if you change the topology, then you will produce OR gates or XOR. The great thing about it, it's when the operation, let's say, sorry, it's a sort of calculation word, but when the process is done, so the promoter was activated once, then it's within the structure of the system. So if it was activated a couple of hours ago, if you just look at the bacteria keep the signal because it's within its own structure. So it's not only something fuzzy and dynamic which occurring, okay, it was increasing and then it's going to decrease. It is going to stick in the genetics. So you can wait. Let's imagine this one occur first, and 1 hour later this one occur. Then at the end you will have the sum of that and that. And you don't have to synchronize, basically. So that's a great thing about it. And basically, if you look at it, that's a principle of a memory. Let's think that's a bit, and that's a sort of 2-bit calculator. You have bit 0, 0. Here you get 0, 1, etc., etc. Okay? And so it's basic of calculation, and it's implemented in the genetic of the bacteria under the control of promoter. So we had them to characterize that, and using this kind of thing, we try to transform that in something we can use in a clinical environment, which is really, really far to be the case right now, because in a laboratory it works, but it doesn't work 100%. You don't want the doctor telling you, oh, you are sick, and then phoning you, oh, no, you are not. Finally, yes, you are sick. You know, you have something reliable, because for many reasons. If it's the flu, it's not a problem. If it's an infection which is going to kill you in few hours, then you have to have a quick decision and reliable. In clinics, that's our problem. So we cannot afford 70% or 80% of working cell and 20% not working. So we have to fix a bit the problem. So we optimize a bit the cell, and we make them working, to make it short, in serum and urine, and not in culture and media. It's not media anymore. It's just in urine and serum. We test different kind of things, but now they are quite happy. And obviously, when you are in such a kind of complex system, there are a lot of things for activating your promoter and inactivating your promoter. There are a lot of molecules in urine and blood, and so you have to, with some trick, to fix that in a genetic system in order to come from this kind of problem to this kind of response, cleaner, much cleaner. Just for the little point here, when we look, we don't look anymore at the tube response, at the fluorescence at the tube, because that's a sum of all the bacteria within the system. We look at this with a cytomate, okay, at individual response of each bacteria. And as you see, we can be happy with a couple of signal here, if you look the sum of all the signal with a given threshold, but you are not happy in the clinical environment when you look at the bacteria. So in order to have a more precise way, and sometimes the result here are really interpreted in a different manner than the result here, we have to go through individual cell measurement in this kind of experiment. And so we did that with the working system with arabinose and hydrotetracycline, and we then shift the promoter for glucose activation of the gene and nitric oxide activation of the gene with this kind of promoter. It's a glucose responsive promoter, and here in nitrate and nitrate sensing specific promoter. So we engineer that in order to have cells looking like for the vesicle earlier, looking at both of them with the end-gate relationship. But if we want to play with them, we are not going to spray the hospital with genetically re-engineering bacteria. They are not going to like that at all. So we have to think about a format, how to use them in order to keep them where we want. And so we made alginate beads in which we trap alive bacteria, modified bacteria, but the compound are able to go through the alginate pore and networks. And so that means now we have beads, alive beads, basically, with bacteria, with the compound negative or positive one, change the fluorescence given the parameters on the environment. And this is very stable and very easy to do. So the beads of alginate are here if you want to. So that's a way, for instance, to work in the real media, like urine and blood, with engineered bacteria looking at 2 parameters. 3 parameters means the complexity is much, much higher. That's what we are working on right now. And to end, I'm showing you the current project with some colleagues in Europe, where, again, the toilet, sorry. Here you got the urine. And what we do is to make our system is perfectly able to capture parameters from the people within urine, even diluted urine, no problem. But now, how are we going to read? You are going to understand why I asked this question. Basically, fortunately, the population is aging more and more each year. We don't want the people with their whole life spending as long as they are age, then they got sick a bit more and more often. We don't want them in the hospital every day. Most of them got chronic problems, big or small. It's not necessarily a problem, but if they are controlled every day, then this is a really good advance and good benefit for the patient. So here the trick is how to keep the people at home and measure the parameter every day without doing anything. Because let's now imagine you are 80, and you are alive, well health, but you have a diabetes since 15 years. When you have diabetes for so long, you have complication for heart, kidney, but as well eyes. You may lose your capacity to see, and sometimes you become blind. But fortunately, let's imagine the capacity to see well is lost. So how the people are going to look at the color of the urine there? So we cannot trust this kind of thing, because there are so many different individual cases that for sure we cannot rely on the people which are going to read at it. So if we need a doctor coming at home and looking at that, it's not possible. So the system is going to read for us, and so we use electronic compounds, which are going to float on the top. So our system, I showed you before, instead of sending fluorescent compound, is going to send chemical compound, exotic one, in big amount when it's positive. And so your urine here is going to be concentrated in a new compound produced by the bacteria or the vesicle. And this chemical compound is going to be captured by MEMS. MEMS, maybe you know, there are electronical compounds which make the panel turning when you move. You know, they are sensitive to gravity, they are sensitive to pressure. There are many, so it's called MEMS. There are nano compounds. It's full of nano compounds, this kind of stuff. And they are very sensitive. If I don't know, I'm not very good with that, but if I turn it, it's turned with that. So they can play as a very tiny little sensitive balance. So we're going to put this kind of MEMS on the top of our capture here, our sensor, and this MEMS is going to be modified with chemistry, able to bind the chemical compound we produced here. Okay, so let's say avidin, biotin, for those which know, or something like that. Then you produce a compound when it's positive, measuring a couple of parameters with our synthetic bacteria, for instance. The compound is going to stick to the MEMS. The MEMS is going to accumulate the molecule, detect then the molecule. How are you going to read that? Exactly like when you stole a CD in a shop. You know, you go, it is ringing because there is another compound which is called RFID. It's a little area, antenna, if you want. And this antenna, when it goes through an electromagnetic field of the shop, it's ringing, right? So we put and we couple this MEMS to this kind of antenna area, and then somewhere here the people will have a new kind of phone, and for sure, listen to me right now, you will see in 5-10 years or less than that, we have a new kind of phone with devices for Earth, and these devices are going to just not even ring, doing nothing basically. It's going to capture the signal and send by internet or by mobile phone the result to your doctor, and the doctor is not going to do anything as well. He's going to talk with the patient, and the computer is going to look at the parameters. If the parameters are high once, okay. If they are high every day, then he's going to ring the doctor and not going to do anything. He's just going to change the schedule of the doctor, in the computer, and the one of the doctor, just to arrange the schedule in between the doctor and the patient, right? So that's kind of science fiction Earth, which is very close to us, right? So it's a research project. There are people from electronics, computing science, electronic signal treatment, health security, clinical, medicine doctors, chemists, biologists, and many other people, and a lot of fun. So that's a kind of current project. Not only that's our project, but there are other projects even more crazier than this one. But at the end, you have to keep in mind that that will be the take-home message, that synthetic biology, there are many, many ways. So we just talk about very few things, but there are, as I showed at the beginning, there are a lot of groups doing different things. It's really boiling area right now. It's not a complicated area. So a lot of people, even students, undergraduate students, there are an international concourse of undergraduate students, which is called IGEM, in the States, so it's fun, okay? But it's just undergraduate students. So modifying a bacteria with bricks, which are already provided through internet, it's very easy. So you have to keep in mind it's not that complicated. The complicated one is what you are going to do with that. Having something working few seconds in front of you, it's great, but you are not going to do something with that. So and robustness in biology is complicated. So you have to go through engineering and open our, let's say, area, which is biology, to something which is close to mathematics, engineer, physician. Go to the coffee and discuss with them. It's very interesting. They can tell us many interesting things, and they can learn from biology too. But the problem is not only so it can help as well to understand biology. I was showing you how we look at the networks and we look at the logic capacity of natural networks. And basically now we discover that in biology, the logic capacity of a network is not homogeneous. There are areas where there is a lot of logic in natural networks, and there are areas where the logic is very poor, and we don't know why. So we learn probably on biology. But it's not, the clinical application, it's more than a technical challenge. There is not only the difficulty of doing the things working robustly and stably, but there is ethic issue. And this ethic issue, if they are not addressed, then we will see nothing working in a real life. Because it's more than a GMO compound. It's created from scratch. It's probably non-pathogen, but you don't know about that. It's probably non-pollutant, but you don't know unless you test it. So you have to address that. And my feeling is asking the question earlier will help the society accumulate, let's say, aggregate this capacity to understand what it is early, and then it's not going to be any fears or fears out of that. But IP issue is something, well, it's quite easy to understand. If you want to have something in real life applicable, so meaning, let's make it short, something you can buy in a supermarket. If you buy something in a supermarket, that means somebody is going to produce it. If somebody is producing it, that meaning somebody spends some time trying to arrange a chain of production and expect making some money out of that. Because they cost a lot to produce something, to transport something. Okay. So if somebody produces something, so that means there is a way to have a market behind. If there is no intellectual property, no patent, then all the concurrent we can do the same. And we can feel, well, that's better, it's free. It's free, but nobody is going to build something complicated to produce it, to transport it, and to sell it. Because there is no business, it's free. So it's very complicated to keep low cost, because the goal here is to be very, very, very low cost. Okay, less than €1, less than $1 the test, even less than that, 10 times less than that. But the point is, if it's zero, then that means no industry are going to produce it, to calibrate it, to standardize it. It's not research work. That's the very beginning what I was saying. It's industrial work. It costs a lot to develop something that you can put on the shelf of a supermarket. So if we don't think about ethics, if we don't think about how to protect it, to bring some value out of it for somebody's hands, not me, somebody else is going to use it, then it will be nothing. It will be a nice publication and nothing in the clinics to use. And again, if we want to clinic use, there are some regulatory issues. It's not legal to put such a kind of thing on the market, but it will in the future. And now we are working with the authorities in FDA, in state, and in Europe to try to figure out what will be the safer way to produce that, work on that, but not putting any kind of thing in the market without looking at it. So the kind of balance in between security, safety, and technical progress for the benefit of the patient. So it's very complicated. There are a lot of different actors, and not only scientific, and the dialogue are difficult sometimes. But I think we need to address that. And when I say we, it's as a scientist, we need to be there in the discussion for ethics, IP, regulatory issue. It's not our specialty, but if we are not there, people are going to decide for scientists. So I think scientists know more or less what they want to do, but they very often are not here, and my feeling is we should be there. So obviously at the end, that make our life very complicated, not only at the bench, not only trying to find some money for doing the experiment, but as well to find some time to support the invention and the experiment in order to be used and useful for the patient later on, and it's a really, really long chain. That's it. Just few words, just to say that obviously I'm not doing that with my little hands myself. So it's this group for synthetic and system biology in the lab which is doing that. Just for the thing, there are chemists, mathematicians, computing people, and biologists. And obviously we work with clinicians, and we have many collaboration because we don't know how to do everything by ourselves. So thank you for your attention.
So, for example, in the system that you were describing to for diabetes, you were trying to build this sort of an AND gate. So, if you want to use it with a patient, that you can do it when he's at the bathroom, detecting glucose, and the other one's going to be detecting nitric oxide. So, being those biochemical reactions that are going to have a Q10 that's going to be maybe 2, or maybe the other one's going to be 2 point something or 3. How are you going to deal with temperature compensation? Because this system is not going to be temperature compensated. So you might,
it's true.
You might set your system to work in the lab at 25 degrees. You might have a different threshold.
So that's a very good question, and that's generally speaking the kind of problem we have, but I didn't show everything. All the problem we have, we are more problems than solution anyway. But this kind of problem there are many like that. So who's going to be sure that's you, which was there, for instance? So we have to set up all these things. So for this specific question, there are different ways to address that. The temperature affects basically the entropy of the systems, and so the dynamics. Not much the quantity, but the dynamics. So the relative quantity, basically the one which is going to eat the interaction. So there are two ways to address that. Although you capture the temperature in the system, and you, thanks to the computing application, and you remodify, because you have tables, and you modify given this temperature, just increase or not the given result. That is basically the position of the electronician engineer. Oh, we know we have a capture with the temperature, we know how to, but that's an additional capture. Remember that we want very low cost. At the end, almost zero, but not zero, okay? But very low cost. So adding that means more complexity for the capture. The other way, the one which I found smarter, but not easier, it's to use memory. Basically, again, the dynamics means things are coming quickly, or quicker, or later. But if you look at the addition of the signal, then at the end you just look at the additional signal, and you look at a sort of quasi-qualitative result of accumulation of it, like a bit Geiger counter for radioactivity. You know, it's a count per minute, for instance. Now it's not a count per minute, but you accumulate it on the memory, and you stick tuck tuck tuck. But when you got a hit, it's stick in a memory, and then you add on the top of it. And then if you, in a given time, you have a sort of threshold which goes over, then you may, if you have obviously a control, I didn't talk about the control, so there are other molecules we are looking at, which is going to be a sort of expectable compound. So this one is going to tell us the system is going quicker, the system is going slower. And so you can arrange that and turn out. But it's not easy because meanings in a network are ranging. That's what do the nature. But the nature is optimized, not the word. The nature is not optimized. Please remind. It's another debate, but the nature is not optimal, right? But the nature works well, and and it's complicated. So then it's more optimal than our system. So. You can think about a balance, a sort of automatic balance, which reduces that automatically by itself, and I would prefer that than only adding new captors. But you can do this two ways.
I have two questions. The first related with your functional paradigm, trying to understand what is happening in the system. What do you think about, for example, the microbiome? Can you measure what is functionally not depending who is doing what?
Yeah.
Not even the cell, even the molecule. You said you only care the domains. So it is possible to interpret what is happening in the system by using your own approach.
I would love. No, I think it's. I see your point, and I think that's a graal where people want to go, because basically we don't care about which molecule did the work, if the work is done at the end of the day. Why do we care which molecule is doing what? It's because we don't know much, and we look at PCs, and PCs is so-called analytical method for discovery, Descartes and so on. The systemic approach means it's not by decomposing a system that we're going to have the system functioning. It's I can't remember the guy. It's an epistemologist which say when a complex system is decomposed, you just remove one piece. It's not anymore the complex system. It's a new one. So the system should be seen as a whole. And then, as you say, doesn't matter who is doing the job is done. And in terms of function, we don't care about the thing. That's our approach. But I tell you, we do that with small networks, and now bigger and bigger, but it's an amazing work. And I cannot say that now that we can manage to do that on a large scale, microbiome, nobody understands anything. In the Congress, I will play a bit with all the literature we found. It's really exciting. And let's say we understand that it is important, but we don't understand anything. And it's great because I was in Cambridge in July with a nice congress on microbiome and pathology with people in Canada. They are very advanced, where they already use probiotics for cancer people to try to accommodate the treatment. And it works very well. And suddenly it doesn't work for this one, he died. Oh, they stopped. Okay, they stopped. They decide to stop. It's forbidden. So, well, unfortunately for the guy, but meaning that it looks like it works, but it doesn't work for everybody because it's not that simple. It's not the microbiome. It's a sort of ecosystem in between. For you, it's easy to understand. For this community, in between the immune systems, the microbiome itself and its parameter, plus the surrounding of all of this. Okay, so we understand that it is very exciting, but part of that, but there are a lot of funny things out of that.
Which is related with the wonderful device, but all related to the biomarkers.
Yeah, that's it.
Glucose and plus.
Okay.
You know, is diabetic for everybody?
No. So I didn't show that, but I would say 70% of the effort of the lab is based on which biomarker. So let's say classical science with systems approach and so on. Because the key is which biomarker look at. And obviously for diabetes it's not enough, but we for instance, we recently discovered biomarker for early renal complication in diabetes. And some other for early heart complications. There are people looking for biomarkers in the blood in our lab for occurring in the blood before the heart attack. And so the game is having again this little mobile phone, which at the end is going to be useful. Right now it's not really, but it's going to be useful. The game is for us and for our concurrents is to identify the biomarker, circulating biomarker in the blood, maybe in urine, and giving the alert on the mobile phone with localization and sending the alert directly. So you do nothing, and the ambulance come. So you can, if it doesn't work, you're gonna have fun. But well, that's the kind of thing. In Mexico right now, people think that's a sort of rich country game. No, it's the opposite. I came from, I was in Hanoi last week, and in Mexico in March. And these people are expecting this kind of thing, because otherwise that means building new hospital for the retired population, which are very big and so on. Now what they are doing in Mexico, very interesting. They are, for pregnant women, they were having a level of pregnant women, accident with pregnant women which were dying because they're coming too late, with too many people at the hospital at the same time. Anyway, so now they design an electronic device to put here, and they are doing comment dire, une échographie, c'est ça? Échographie. An échographie, a small one, without sound, nothing there, and connected with the phone. And the echography 3 times a day. 3 times a day, the girl have an echography. It's never reached in any kind of country. Small one, okay? And when the heart is not sounding anymore, the system is doing nothing, because otherwise the mother is going to shout, cry, maybe fall down, running after the phone. Okay, so it's doing nothing. It's even producing the sound of the heart, because they realized that the mother was very motivated to listen at the heart of the baby. So otherwise they are not putting the system. So they put the heart of the baby, and they listen to the heart of the baby. If there is no heart sound, then they still have a heart noise, which is replacing the right one, and they give the alert, and the ambulance come. It is working right now in Mexico, and it's not a high technology stuff. Huh? It's just doing the stuff. But I asked them the killing question: what's happened if the network is not working? Well, we got a problem. So that's why in the future, you have to know that, even if it's not our field, but in the future, all the telecom company for every country are thinking about creating specific network, mobile network, for, let's say, security and safe application. Civil application for safety, regulatory stuff, and the kind of Earth, something secure on which you can rely 24 hours a day, and it works. But it's heavy, it's expensive, it's long. But just to say that we are going this way, and not only in Europe, on the States, on Japan, or in South America, but everywhere. Muchas gracias, doctor Aguiliani. Gracias. Combien c'était sur le temps?